Hi, thanks for joining us again today for episode number 91 of the Doctor Podcast Psychert File Show. We really appreciate your watching today. And we also would appreciate it if you could subscribe and follow Doctor Podcasts and also like and share and repost this episode so that everybody else can see it as well and we can get additional great guests like we have today. Today, we're going to be discussing a very, very important topic, and that topic is mirror life. And this could be an existential uh, condition for the human race. And just listen to this, and you'll find out why. Our guest today is the esteemed Dr. Jack Szostak. Dr. Szostak won the Nobel Prize in Medicine and Physiology in 2009 for his work on telomeres and telomerase, and we'll hear some more about that in a few moments. He's a university professor at the University of Chicago, which is freezing today, and he heads the Szostak Lab and is director of the Center for Origins of Life lab, which is researching the chemical and physical processes that facilitate the transition from chemical evolution to biological evolution in early Earth several billion years ago. Previously, he was a professor of chemistry, genetics, molecular biology at Harvard Medical School for many years as well. Now, in addition to the Nobel Prize, he's also received numerous honors and awards over the years, has published numerous seminal articles on biochemistry, genetics, and the biophysics of life. His research has recently ventured into the fascinating realm of mirror life, which explains life forms with a mirror image biochemistry. Um, and most importantly, he recently co-authored in December a publication with 37 other authors, many of whom had Nobel Prizes as well, warning of the risks of mirror life research. And this was published in the Science Journal, a very prestigious scientific journal. So, Jack, thanks for taking the time today to join us. We really appreciate it. It's a very important topic to discuss.
Well, thanks for having me on this.
Yeah.
Thanks.
So tell us first about your Nobel Prize, about your telomere and telomerase research and how that led to winning the Nobel Prize. Tell us about that.
Sure. That's a long time ago. uh, Back in 1980, I think it was. I heard Liz Blackburn gave a fantastic talk at a Gordon conference on nucleic acids. And I went up to her afterwards. We had a great conversation and we decided to collaborate. She had been working on telomeres from an unusual organism called Tetrahymena. It was very strange cell biology. And yeah, we thought it would be interesting to see if the telomeres that she had isolated from Tetrahymena would also work in yeast, which is the organism I was using as a model at the time. And neither of us thought it would work, but we thought it was worth a try because if it did work, it would say that the underlying biochemistry is very highly conserved. And so it did. It did work. It led to several years of experiments in my lab, although I have to say it was never the main thing we were working on. My lab was focusing on DNA recombination and repair, among other things. But it was, yeah, it was a pretty good side project to have going for 6 or 7 years.
Right. And eventually it led to your winning the Nobel Prize in 1999. So,
Age nine, is that right? No, no, 2009, sorry.
oh,
2009
2009.
got the Nobel Prize.
Okay.
And the reason why it took so long was that at the time of our work, it was just, we were just happy to have solved the longstanding puzzle in biochemistry of DNA replication. And then, later on, it turned out to have really important implications for both cancer and aging, which lots and lots of people are still working on.
Right. Apparently, if you have long telomeres on the ends of your chromosomes, that's a good sign that you're healthy and have good longevity. Is that right?
It's a double-edged sword because if telomeres are too long and in the wrong cells at the wrong time, that means that if a tumor starts to develop and the cells are continuing to grow and divide and divide and divide, they don't stop growing when they should, right? Most tumors die out because they're the telomeres of those cells get too short and the cells die. But if telomeres are too long, then
Hmm.
it's not good.
So you have to have just the right size. So tell us about the morning you found out that you won the Nobel Prize. How did that happen? What were you doing when you found out, and were you surprised about that?
So at the time, I was still at Mass General and Harvard, and so in Boston. Um, so typically, I think the phone call from Stockholm comes at around 4 in the morning.
4 in the morning. Great.
And you have about an hour to get up and get dressed before reporters start knocking on the door. And then your day is chaos, and most of the following year is pretty chaotic too. But it was exciting, it's just
Right.
great to feel that your colleagues, your peers, really like something you've done.
Right. It's the highest accomplishment in science and in medicine and physiology. So congratulations on that again.
Thank
So
you.
when did you become interested in chemical to biologic evolution of life? I've always heard the theory that a few billion years ago there were a bunch of chemicals swimming around a pond somewhere on Earth, and a lightning bolt somehow hit it, and then the first cell was born, and we've evolved from that over the last 2 billion years to now be doing video podcasts. So tell us about that theory and how you're working on that.
Yeah, sure, sure. So, yeah, I started to get interested in that about, what, 25 years ago, I'd say. And that interest came out of the work I'd been doing the previous 10 years on RNA biochemistry and ribozymes and how you could get RNA molecules, find RNA molecules that could catalyze different kinds of chemical reactions. And in doing that work, we were basically learning how to do molecular evolution, how to do evolution with molecules instead of with organisms, just doing it with populations of molecules. And that was very interesting for a long time. Um, but after doing that for a decade or so, I got more and more obsessed with the question of how life itself, which is, how did evolution get started all by itself, right? I mean, when we're doing these things in the lab, we have, every modern instrument, all kinds of analytical processes at our disposal. We have brilliant graduate students who can actually do the experiments. Uh, but somehow life got started all by itself, which means there was a path going from the chemistry of the early Earth to the beginnings of biology. Once you have biology, which means you have a Darwinian evolution actually happening, from there on, evolution just takes you forward and you have organisms appearing that are adapted to different environmental niches and they're competing with each other and they get more and more complicated so that they compete better and as you said, here we are.
Right.
So it's that transition from chemistry to biology that's really something I've been really focused on for the last 20, 25 years.
Wow. So it doesn't happen overnight. So have you figured out how that transition happened? Is it evolution, or do you believe possibly in the panspermia theory, where some meteor or comet crashed into Earth from another planet or perhaps another solar system and just delivered a bunch of proteins, RNA, DNA, carbohydrates, and that's how it started?
No, I think it all happened here on Earth. Um, Meteorites are constantly crashing into our planet and they deliver tiny amounts of organic compounds, but what they deliver is a huge mess of, thousands or millions of compounds. It's not what you need to start life, right? What you need to get life going is you want to have a lot of the right compounds.
Mm,
And so that's a question in chemistry, for which a huge amount of progress has been made recently, largely from the lab of John Sutherland in the UK and also many of his students and colleagues over the years. So we we in the field have a better and better understanding of what molecules could have been available, what sources of energy were required and available. And the part of the puzzle that I've been focusing on is once you have all of that, then what? How do a bunch of chemicals get together and start acting like a living cell?
right,
Uh, and, like any big problem, we try to break it down into smaller, more manageable problems. And they're not that manageable because pretty complicated questions.
right,
But it's fairly simple. If you think of modern cells, they have a cell membrane. It's made of lipids. There must have been something like that for the first cell, probably much simpler membranes, much simpler molecules. So that's one half of the overall puzzle we've been working on. We've made a lot of progress there. We know how to make those vesicles, so protocell membranes. We know how to make them grow. We know how to make them divide. We know how to get nutrients in. Uh, so there's been a lot of progress there. We're not finished on that by any means, but a lot of progress. And the other part, which is even harder, is you need something like RNA to carry genetic information, to allow evolution to happen, and also to catalyze reactions, right? So like Tom Cech and Sid Altman discovered years ago, RNA can also act like an enzyme. That in itself was a huge breakthrough in thinking about how life got started. And so our focus has been on how do you go from the building blocks of RNA, which are nucleotides, to oligonucleotides, short strings of RNA, to longer RNAs. How could that process happen? And the hardest part, how could those molecules replicate so that if there's a good one, it can be propagated and evolution can start on. So again, that overall thing gets broken down into lots of sub-problems, and some are solved, some we're working on, some we have theories, but still a long way to go. And the ultimate idea is once we can replicate RNA without enzymes, without evolved machinery, we would like to have that inside our membrane protocells and have the whole thing growing and dividing, replicating its genome and evolving new functions.
so you're basically trying to reproduce a few 100 million years of evolution in your lab in a few decades.
I don't think it's that bad because these molecules are delicate molecules. In addition to being made by simple chemistry, they also get degraded by simple chemistry. So I think on the early Earth, it may have taken geological time, tens of millions of years, to set the right environment up. And then maybe it took, in the right environment, I don't know, thousands of years to accumulate a reservoir of the right starting materials. And then once they're processed and you have really reactive compounds and they start reacting and building up the starting materials for biology, that's something that probably happened on a time scale of, hours, days, and weeks.
Mm.
And then once you have a first cell that can grow and divide, that's probably happening on a time scale of hours and days. So I think the critical pieces happen on fairly short time scales. It's just setting the stage and getting all the parts together that might be what took a long time.
Wow. So, how close are you to creating a life form in the lab that can reproduce itself?
Well, for the last 20 years, I've been saying I think we're only a few years away. And there's always new problems that turn up, and then we solve them. We think we're close, and then some new problem turns up. But yeah, I still think actually we might get there in the next 2 or 3 years.
2 or 3. Wow. Now,
We'll see.
I'm sure there are other labs working on this. Are they kind of in the same range as you are? They all think it's a few years down the road, or is anybody really close? Do you know?
Well, it's an interesting situation. There are definitely lots of other labs working on pieces of the puzzle. Um, I think most people actually think that this is such a hard problem that they don't want to do it. They don't want to work on it. And also, the other thing that maybe scares people off is that the only reason to do this is curiosity, right? We want to understand how we got here. It doesn't have any practical applications at all, as far as I can see. So that
Really?
itself makes it a little bit difficult. I'm very
Uh,
lucky to be in the position that I'm in, that I actually can devote my lab to this work.
Right. Now, if you were to produce this organism that reproduces, let's say in about 3 years or so, is there a chance that this could be a dangerous organism that could leak out of your lab by accident, or maybe on purpose by some rogue, crazy person and cause lots of damage,
Yeah,
potentially be an existential threat to humans and other species?
right. No, I don't think so.
No.
Um, what we're trying to build is such a primitive state of life and so dependent on us to supply all of the building blocks, which are not found in nature anymore. So there's no way that it could uh, propagate outside of the lab and outside of our uh, determined efforts to keep it going. So I think there are no dangers like that.
You don't think so?
No,
But what if, let's say, you produced this organism and somebody in your lab inhaled it and, got sick from it and then went
they,
out
they
and
wouldn't. They would just digest it. RNA is so delicate; it would just get
wouldn't
digested.
happen.
The membrane's made of simple things like fatty acids that we eat all the time. So yeah, it
All
would
right.
just get digested.
All right. So let's talk about mirror life now, which is
Okay.
the subject
Okay.
of this paper that you co-authored about a month ago. So life on Earth is a mixture basically of proteins, carbohydrates, some lipids. The proteins are made out of what's called
And the nucleic
left-handed.
acids. Don't forget the
And you,
nucleic
yeah,
acids.
I was
RNA, DNA, right?
won't forget those. Very
Central.
important, right? And basically, the proteins are made out of left-handed amino acids, right? And the carbohydrates are made out of right-handed sugar molecules or glucose. And then DNA and RNA is also right-handed. So why does life on Earth have this predominant use of this molecule chirality or right-handedness or left-handedness?
Yeah.
And what theories exist about why this emerged and why it stayed that way?
Yeah, sure, sure. All very interesting questions. So if you look at the building blocks of RNA and DNA and proteins, um, most of them are they're relatively simple molecules, but they're complicated enough that they have they're not identical to their mirror image, right? So it's like your right hand and your left hand. You can't superimpose them, right? And uh, all of the sugars, nucleotides, and amino acids are like that, except the simplest amino acid, glycine, which is um, it it has no chiral center, so it is the same as its mirror image. But all the other 19 amino acids found in proteins are a little bit more complicated. So they are chiral, which is just the word we mean to say that it's not identical to its mirror image. Now, when you start to make a polymer like RNA or DNA out of building blocks that are chiral, then the whole long chain is it's like a chain of these molecules that have the same chirality, the same mirror image form. And you'll recall from your biochemistry that DNA is a double helix. You have two strands. They have to pair by base pairing, right? And if the other strand has the wrong chirality, it can't base pair. The things don't fit together properly, right?
Right.
Uh, so life, at least life as we know it, life using RNA and DNA as genetic material, has to be chiral. It has to be what we call, say, as homochiral, right? All of the nucleotides have to have this be of the same mirror image form. Otherwise, the polymer won't function, it won't be able to replicate, and so on. And for proteins, it's the same thing. If you were going along, with an L amino acid, L, 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 and then the D one, the D amino acid, then the chain's going to go off in the wrong direction instead of the direction that it's supposed to go in. So you couldn't make enzymes if you had a mix of RNA of L and D amino acids. So one of the questions that's puzzled people for the last 70 years is how did life get started if the building blocks had to all have the correct chirality? And there's been many theories, some that are just crazy and some that seemed interesting, some that if you have an initial imbalance, there's ways to enrich that. There is some very new work that provides what I think is probably the right answer to the puzzle, and it's because homochirality comes into the system at the right point on the pathway from the simplest building blocks up to nucleotides. There's a common precursor in there that is chiral, and if you can make it in only one chirality and only one form, then you could make all the nucleotides with the right chirality. You could make your RNA with the right chirality. So the research in that direction, I think, has progressed very, very nicely. And so now we have all life on the planet, right, uses the same chirality of its nucleic acids and proteins. And so the question now
That's
is
that's humans, all animal species, all plant species, bacteria, fungi, etc.
everything. Yeah. Yeah.
Viruses as well.
And viruses, right? Because we come from one common ancestor.
Right.
And that common ancestor, sometimes referred to as LUCA, right? The last universal common ancestor. That organism was pretty much like a modern bacterium, had all the pieces. It itself had evolved over probably hundreds of millions of years from the very first cells. So I think the logical way to think of it is that, homochirality emerged as a property of the chemistry, that chemistry led to the first cells, uh, which had the chirality that we have today, and it's been propagated down through time ever since. Now, you could say, well, why do we have the chirality that we have, right? We all think that life would work just as well with the opposite if everything had the chirality switched. I mean, everything that we know about physics says that is true. And so the consequence of that is that most likely the chirality that we have in modern life is arbitrary. Could have been the other way around. It wouldn't make any difference. It started one way, and that's just what we got.
Kind of like matter and antimatter. For some reason, matter predominates, at least where we
Right.
are in our observable universe for now, I think. So, It just
So there's
started
an
that way.
interesting twist. There's an interesting twist there. So at the level of fundamental subatomic particle physics, there is a fundamental asymmetry to the universe, which is connected somehow with why we have matter and not antimatter. But at the level of atoms and molecules, the energetic difference between those is so small that it doesn't affect anything practical in our world. So despite that fundamental underlying asymmetry in the universe, the asymmetry of the molecules of life seems to be basically arbitrary.
Hmm. Have there been any cases of mutant chirality or something that dies off, or nobody's ever found that? It just doesn't happen. Even the mutations can't happen that way.
Mutations can't do it because you would have to change so many things altogether, right? You would have to change all of the metabolites, all of the proteins, all of the RNA and DNA. You couldn't, a cell couldn't function with a mixed system. And so there's no way to get to from where we are to a mirror system by evolution. Could only be done by design.
Right. So now are scientists like you and other labs working on creating biomolecules of the opposite chirality in their labs?
So there's a lot of different strands of work that touch on that. The the one small piece of that we're working on actually goes back to work that my colleague Jerry Joyce did when he was a graduate student, I think 40 odd years ago. And Jerry actually showed that if you have a mixture of right and left-handed nucleotides, you can't copy RNA chemically. Okay. And at the time, the techniques for understanding why that were not as well developed as they are today. And so we're going back and repeating some of the experiments that Jerry did and trying to get a more detailed picture of exactly what goes wrong when you have a mixture of right and left-handed nucleotides. Now, there is also has been work going on for quite a long time in making mirror image proteins and mirror image RNAs, RNAs that are called aptamers. They're RNA molecules they're kind of like antibodies. They bind things, right? And people have been doing this for therapeutic reasons um, because mirror image molecules like this are pretty much invisible to the immune system. And so that means if you say you're treating somebody with a therapeutic protein or RNA, it won't get degraded. There's nothing in your body that can degrade these things. Okay, so that's for therapeutic, that can be good, right? Because it would last longer. Uh, and you can make these molecules up to a certain size just by chemical synthesis. Right. So that's the practical application of mirror biomolecules, and people have been doing this for quite a while now. And
Right.
there's no danger with that because those molecules can't replicate.
They can. Okay. Now, I've read that you can create the opposite chirality glucose, for example, molecule, which apparently would have the same sweetness, but would not be metabolized by the body. And that might be the best diet food. You could eat cake and ice cream all day and not gain an ounce because your body can't metabolize it. Why hasn't that been done? Or has
I,
it?
I'm not sure about this, but I don't know, and I actually doubt that it would have the same sweetness, right? Because the
Mm,
sweetness of sugar, and the one that we like the most usually is sucrose or sometimes fructose and glucose. The sweetness comes from its interaction with the protein receptor in your taste buds. And that protein is a chiral molecule, and it
Yes.
recognizes chiral glucose, and so it probably won't recognize the glucose of the opposite chirality. So,
Interesting. So
yeah,
the glucose mirror image molecule would not be sweet, and therefore there's no use for it.
probably. I actually I don't know if that experiment has been done. That would be actually would be interesting.
Right.
It's probably been done.
Right. That would be better than Ozempic,
Yeah, yeah, I don't know.
maybe. Yeah.
Yeah,
So I want to talk about this article that you
yeah.
published. It's in the December Science Journal, December 2024, and I think there were 37 or 38 co-authors with you, several of whom were also Nobel Prize winners.
Yes.
And in the article, you warned about the risks of mirror life. So can you tell us what those risks are,
Yeah,
why you decided to write this article now and not several years ago, and what led you to write this
right,
article?
right. So there has been some recognition that mirror life could pose substantial risks actually, for a long time. There was a very brief article that was also published in Science about 20 years ago that raised some of these issues. And there's been a few other public articles since. But none of those articles really went into detail and analyzed the risks in depth. And that's what our collection of co-authors has been trying to do over the last couple of years. Uh, I got drawn into this work only about a year ago. And I was very skeptical at the beginning for the same reasons most people are, but the more I thought about it, the more I read and learned, the more I came to agree that, there are very, very substantial risks to human health, animals, plants, consequences to microbial ecology. So we can discuss all of those in detail. But the reason why this has come to the fore now is that the possibility of actually creating mirror life is getting closer because technology is advancing very
Right.
rapidly. Right? There are a lot of people who are trying to take, say, a simple bacterium, basically take it apart into pieces, have all the purified components of a simple living bacterial cell, and try to understand how you would put those back together to recreate a living bacterium. And that's a very interesting and important line of research because it will help us to understand how living cells actually work. Uh, the idea of reconstituting specific activities of cells has been the driving force, the core of biochemistry for over 50 years, right? People started off purifying enzymes and trying to understand how they work, and then purifying complexes more of multiple proteins and trying to understand how they assemble, how they work. And now people are getting closer and closer to understanding how to reconstitute whole ribosomes, which are really complicated molecular machines that are what make proteins, right? And the ultimate extension of that reconstitution would be able to take all of the purified parts of a living cell and understand how to put them back together. And we're going to learn a lot, I think, from that kind of work. But at the same time that that's been going on, the technology for synthesizing mirror components of life has also been accelerating quite quickly to the point that it's now possible to chemically synthesize RNA molecules and DNA molecules that are 1,000 nucleotides long enough, in fact, to encode the RNA components of the ribosome.
Hmm.
And it's been possible. This is work done in several labs, but I think most prominently by my former student Tim Zhu, who's now in China. What Ting has been able to do is to uh, chemically synthesize mirror proteins that are enzymes that can copy and replicate DNA, can transcribe DNA into RNA. So you can see that the capability to generate by synthesis all of the components of a cell is getting closer, right? Now, it's not really close, which is I think is a good thing because even the simplest, reasonably robust bacterial cell has a genome of about 1 million base pairs, right? A megabase. And the biggest RNAs and DNAs that have been chemically synthesized and can be replicated are about 1,000th of that. So we have to get 1,000-fold better. How long would that take? Nobody knows, but some people say it might take 10 years or 30, or it could be really hard, or it could turn out to be actually not that hard. So there's a lot of debate, or I should say disagreement, about how far away this ability to create mirror life actually is. It's definitely not just around the corner, but we don't really know how far off in the future this is. And I think it's a good time. We say, it gives us a chance to think about the issues now before the problem is on our doorstep.
Right. Now, you mentioned earlier that you think your lab is about 3 years away from creating a life form from chemicals. If the doctor you mentioned, who was your student, is able to create mirror proteins and mirror nucleotides and so on, then he can put together a bacteria that's made out of a mirror image of what currently exists. Is that right?
Well, in principle, if he kept going and and was able to get maybe 1,000 times better at it, I would say yes. But actually, Ting has become a little bit, and this is probably a good thing, a little bit discouraged with how hard that problem is. And he's actually decided, for probably a variety of reasons, to work on other things. I think he might like to get to the point of making a mirror ribosome, but he's said that he doesn't want to go any further than that.
Right. So let's say somebody could create this mirror bacteria or maybe even a mirror virus. What are the risks of that to humans?
Well, let's talk about the simple thing first, a mirror virus.
Yeah.
A mirror virus would be completely useless, right? It couldn't infect a cell. So viruses that infect bacteria are called bacteriophage. But if you had a mirror bacteriophage, it couldn't infect a normal chirality cell. So the only way to grow a mirror bacteriophage is by first having mirror bacterial cells that it can
Hmm.
grow in. Okay, so we don't have to worry about mirror viruses. But the problem with mirror bacterial cells is that we believe they would be largely invisible to our immune systems, to both the innate branch of the immune system and the adaptive immune system.
Hmm.
So that means that even though they might grow very slowly, they wouldn't be adapted to be good pathogens, but they might still grow very slowly in certain spaces in your body, but they wouldn't die, right? If they're not being found and killed by the immune system. And if that is correct, then they could just keep growing and accumulating and, until they kill you.
Right. It's kind of similar to the TB organism, tuberculosis, which I just had a podcast about, where it just kind of hides there and hibernates and grows very, very slowly for a long period of time. And eventually it can make you really sick or die. So you're saying these mirror bacteria could do something similar.
Yeah, I think I mean it's actually a good comparison because the TB bacterium goes to a huge amount of effort. It's a huge amount of evolved machinery to shield itself from detection by the immune system. Um, Whereas mirror bacterium would just have that from the beginning as a given, right? Its proteins wouldn't interact. They couldn't be digested.
Hmm. So none of our enzymes or antibodies or anything like that would have any effect on this bacteria.
That's the idea. Now,
But
and I think that's basically
how would it get its nutrients if all the
right.
nutrients in the human body are the opposite chirality?
Yeah, that's a very good point. And the answer is that not all nutrients are chiral. There are simple molecules like glycerol, glycine. There's a list of others that are not chiral that it could consume. Now, unless it was engineered to do so, it couldn't, for example, take up normal glucose and metabolize it. But engineering that in, if somebody wanted to, wouldn't be that hard. So yeah, so they would grow much more slowly, but they would also die more slowly. That's the crux of the problem.
Right. Now, potentially these bacteria could spread from person to person, and there would be no way to stop them because our immune systems would not recognize them at all as foreigners. And also, they would be immune to antibiotics and things we use now to kill bacteria. Is that right?
They would be immune to most antibiotics. There's a list of antibiotics that are not chiral that, you know, would still have some effectiveness. I mean, they're not the greatest antibiotics, but yeah, people like to think about what could we do to protect ourselves, if something like this happens. Um, you could you could also chemically synthesize the mirror image of the best antibiotics we have, right? So if there was an outbreak of infections of mirror bacterium, you could protect a small number of people, but you couldn't deploy it on a global scale, right? I mean, I think that's our real concern is that there's no defense that could be deployed on a global scale, and especially when you're talking about not just people, but animals and plants and also
Right.
the ecology of the oceans, for example.
Right. So if you could create a staphylococcus or a staph bacteria that colonizes our skin, is all over our body, basically, that could spread very rapidly and cause lots of damage to millions or billions of humans.
Right.
And we'd have no way to deal with that effectively and rapidly, basically, right?
That's the concern. That's the concern. Yeah.
Could lead to extinction of humans and other species if that spreads around. And then maybe evolution starts all over again with the opposite chirality. And 2 billion years from now, we have two other people like us talking about it.
Or it could, evolution would, keep going, right? And maybe in the end, we would have a mix of both chiralities of life on the planet.
Right,
But the transition to that stage would not be pleasant. Now,
right.
I should say something else, just so, people don't start panicking,
Yeah,
which is that, the first if somebody really tried to create a mirror bacterium, the first one that they would make would not be very robust. And it would be, if they had any sense at all, it would contain, it would be built with what we call biocontainment measures, right? You couldn't design it so that it could only grow if you feed it certain nutrients that aren't even, say, found in nature. And so that way it's escape from the lab into the environment. We think could be rendered at least extremely difficult. And I never want to say impossible because, it's hard to anticipate every action. But the real concern with this is that once you have a mirror bacterium, no matter how simple, no matter how well contained. Engineering it to be more robust, to lose those containment measures, and to be able to grow in the wild would not be that difficult. Now, you might want to say, well, maybe, hopefully, no one would ever want to do that, but, you know, is it worth taking the risk? I think the safest approach is just to not make mirror bacteria in the first place, so we don't have to worry about escape or malicious engineering or anything else like that.
Right. We're always assuming that the world is made out of nice people who wouldn't do anything like that, but we know that's not true. There are a lot of
Right.
evil people out there. There's also rogue people who hack into computers and do all kinds of nasty and vicious stuff for various reasons. So that's why you wrote this article. We shouldn't go
Exactly.
there to begin with.
Exactly. Right.
Now, you mentioned earlier curiosity is a lot of what your work is based on. You're just curious and want to learn more and more, which
Right.
is a very good goal. But
Normally,
are
yes.
there, what's that?
Normally, yes. I
Right.
think, you know,
Uh,
I think we all recognize that there are, uh, in many aspects of science, right? There are technologies that, at the very least, have potentially dangerous applications, and we have to be aware of that, and we have to do whatever we can to be responsible about the development and use of technology.
right. That goes for, the atomic and hydrogen bombs. And now we're facing the same dilemma with AI. Where is that going to go? Is AI going to get us before the mirror image bacteria get us? Is the question.
We have a lot of potential serious problems to worry about. Yeah.
Now, is there any practical application of doing this research to harness important discoveries in pharmaceuticals or biotechnology, or not really?
I would say not really, right? The application that people sometimes talk about is if you had mirror bacteria, you could use them as a sort of biofactory to make mirror image proteins. And you, as we were talking about before, a mirror image protein could be used as a therapeutic, right? And the same thing for, say, mirror image aptamers. But you can chemically synthesize those molecules, right? You don't have to create mirror bacteria to create molecules like that. I mean, it might be easier and cheaper, but I would say it's not worth the risk.
Right. So the article emphatically says, just don't do this. Stay away from it.
That is our consensus.
Right.
Now, we're acutely aware that we're just a small group of scientists who've been thinking about this. This isn't something we can impose on the world, right? We need, as the next step, to have more people discussing this. We need to talk to, regulatory agencies, governments. Uh, we need to talk to the broader scientific community and to the public at large, so that people understand uh, that this is just something we should all agree should not be done. Right?
Right.
And then the hard part is how do you actually implement, how do you devise policies and implement regulations that will hopefully be effective. So that's going to take a lot of discussion with a much broader range of people.
Right. We haven't been able to do that with AI. There was talk a couple of years ago of having regulations and laws and rules, and nobody could agree on how to do that. And we were always worried that some other country is going to come up with a better AI quicker. So there's
Right.
still a huge AI race going on. Now, in your article, there was one sentence that I was curious about. It said, I'm quoting, we also recommend research to better understand and prepare for risks from mirror bacteria, as
Yes.
long as neither mirror bacteria nor any key enabling precursors are produced.
Right. Right.
Um, so once you produce the precursors, which from what you're telling me, several labs are working on, there's a temptation to mix those things together and create the bacteria. So isn't that a bit of a contradiction?
So what we would what we think um, it should be a sort of a red line that shouldn't be crossed would be to make, for example, a mirror image genome, right? Which then, if you had the other pieces, could be used to generate all the mirror image RNAs and proteins. So we think that's an enabling technology that should not be reached. Um, but there are a lot of things well short of that that could be done to give us a deeper understanding of the risks, right? A lot of what we said about immunology, microbial ecology, plants, it's I think it's very reasonable and it's supported by a certain amount of evidence, but it would be good, I think, to do simple experiments that would validate our predictions, short of, not involving making rare bacteria. But for
Right.
example, you could do things like making little beads the size of bacteria that are coated with mirror proteins, right? And you could see, how does the immune system respond to something like that? How do um, microbial predators respond? Can they, or macrophages, for example, can they track such beads and engulf them? What happens? There's experiments like that that I think could be done that would give us a much more detailed and convincing argument, if they turn out the way we think, that these risks are real.
Right.
And I think everybody would be very happy if those experiments were done and it turns out, oh, actually, wait a minute, the immune system's perfectly capable of dealing with this. Then we can all relax. I
That would
don't think
be
that'll be.
That would be nice. But this kind of reminds me of the gain of function research that's been done with the viruses, the
Right.
coronavirus.
Right.
There's some of that going on with the
Sure.
H5N1 flu virus. And, the scientists who do this all claim, well, we need to learn about this so that if we ever have an outbreak of the nasty flu virus, we know how to deal with it and how to create a vaccine for it. But it actually doesn't work that way because it seems like it's much easier to create the vicious, horrible flu virus, and then it takes years to develop a vaccine for it. So isn't it just better to not try to create the monster?
Yeah, I think that's a very interesting and difficult question. Um, the ability to generate effective vaccines nowadays is much faster than it used to be. The mRNA vaccines are a revolution in vaccine technology, and the development of the mRNA vaccine against COVID probably saved millions and millions of lives, despite what people like RFK Jr. say. I
Okay.
don't know if we want to go there, but
Well, there are some people who, disagree on that. So that's a
yeah,
long debate.
it's a long debate, right?
We won't be able to settle it during this podcast, but
Yeah, I think the ability to create vaccines. I mean that COVID mRNA vaccine was designed in a matter of weeks, and now that the production technology is there, I think things could be done much faster. So it's always good to be prepared, but you want to be prepared in a way that doesn't cause the problem that you're trying to solve. Yeah.
right. Now, I'm just curious, were there other people who were contacted to become co-authors of this paper that you published
Mhm.
who either disagreed with your take of not making the mirror image microbes or just didn't want to sign on to this?
Well, the most uh, So not a lot. M Most of the people that we've talked to eventually, agreed that this is a problem, and there were only a few people that we had hoped would be co-authors who declined. And I would say the most common reason is either they didn't know enough about it, didn't want to make a judgment, or felt that the problem was so distant that it's not something that we needed to be concerned about right now. And that's okay. People can have different opinions about these things.
Right.
As I was saying before, we think it's good to deal with these issues now before the problem is imminent. Uh,
Have
but
you been contacted by any government agencies about this to start talking about it and maybe implementing some rules and regulations and ways to verify that there aren't some rogue labs or rogue scientists that are working on this?
yeah, we've had initial conversations with different governmental agencies in this country and a few other countries. That's a long-term effort. Um, the reactions, I think, in general, have been one that this is something to be concerned about, and I think the easiest step that so far everyone we've talked to has agreed on is just to not fund work that is specifically aimed at creating mirror bacteria. So for example, some time ago, I don't know, 10 years ago, roughly, there was actually a small an NSF grant given to a group of people, including some of our co-authors, to try to do this.
Really?
Right. And of course, they didn't get there. they, they, they start to realize how hard it would be. Um, but all of those people have since said, look, now we realize this would be a really bad thing. And so we're not going to do it. And I think the NSF will not fund it in the future. And I think one thing we would like is to have funding agencies worldwide not fund work that's explicitly aimed at creating mirror bacteria.
So the NIH should not sponsor or fund
Right,
any of this type of research.
right, right.
All right. That's important to know.
Yeah.
I have a theoretical question. You know, Elon Musk is out to get to Mars, and he says in 4 years we'll have humans on Mars. Is it
Mhm.
possible that there could be some mirror bacteria or other life forms on Mars that evolve differently, the opposite chirality, and then we could pick some of those up and accidentally bring them back to Earth?
So it's a theoretical concern. And in fact, NASA has been thinking about this problem for a long time, it turns out. What if, some large meteorite from a different solar system crashes into us bearing mirror bacteria? I mean, I think to me, the fact that we don't have mirror bacteria around, it's never happened, means that in 4 billion years, nothing bearing mirror bacteria has come to Earth and survived. And we know, for example, for Mars, there's been a huge amount of exchange of material between Earth and Mars due to large impacts, over the past 4 billion years. And so I think that means that if there is life on Mars, it's probably not opposite chirality. So I don't see that as a serious risk.
Right. On that subject that you mentioned about the meteor crashing, your colleague from Harvard, you may know him,
Avi
Dr.
Loeb.
Avi Loeb. Yeah,
Yes, I know
I've
Avi.
yeah, I've interviewed him a couple of times
Yes.
about this topic. He believes that there are two meteors that crashed into the Pacific, IM-1 and IM-2, and he's actually had an expedition of IM-1 and picked up some materials from there. It's possible that that could be interstellar from his calculations and from what the government said. And potentially, he thinks there may be some form of alien technology in those meteors. And, maybe if they're buried in the deep Pacific, those mirror bacteria or organisms won't leak out to the rest of us. But that is a theoretical possibility, don't you think?
Yeah.
Especially if it's from another solar system where
So,
evolution
so,
is
so the
different.
thing with Avi is that when there's some interesting, maybe surprising event that happens, he will always go and publicize the most spectacular and least likely potential explanation. And it drives all of us crazy, to tell the truth. I wish he wouldn't do that. And I
Well,
think
he has
little,
some pretty solid evidence. He's done an analysis of the little tiny spherules that he got from the meteor.
yeah. So yeah, it's great. It's great to do that. It's great to get more observations. I don't think anything he's found goes any way at all to suggesting alien technology. I think that is just the
So
most
far, not. That's correct.
outlandish explanation, right? So, yeah, well, I don't know why anyone would always try to get a lot of publicity for a low likelihood explanation.
It's a theoretical possibility. He's mounting another expedition to get bigger chunks of this meteor so that we can get more information about it.
Yeah, very good. I wish him
Hopefully,
good luck with
hopefully,
that.
what's that?
I wish him the best in that endeavor.
Okay. Um, we'll see what happens. But if indeed it did come from another solar system, there's a remote possibility that evolution occurred differently there. So, but so
Sure.
far,
I mean, if we find life from a different solar system, right? I think the chance is 50-50 that it will be the same or opposite chirality as ours. And if there is
right.
life on a different solar system with opposite chirality, we would obviously have to be pretty darn careful. Fortunately, the nearest solar system that has a rocky planet in the habitable zone is around Proxima Centauri, which is, 4-ish light years away. And the nearest solar system with an Earth-like planet around us orbiting a sun-like star is considerably further. So again, we're not going to have to worry about this for a long, long time.
Hopefully not, but
Right.
prizes occasionally,
Occasionally
occur.
happen. Yes.
So,
Yeah.
so we
But
have to.
if it hasn't happened in 4 billion years, you know, how worried can you be?
Well, that's true. Or there's a possibility that we just don't know it happened because we haven't discovered it. And so, it's like before Columbus set out, they used to think it was only, Europe and Africa.
Right. Right.
And then
Yeah.
they figured,
But like, so so exploring these other worlds around other stars is something that we will do eventually, but the technology to get there is a long ways off. And unless we learn how to do faster than light travel, which may never happen, then getting there is always going to take thousands of years.
all right,
Uh,
so as long
and
as, sorry, go ahead.
I was just going to say that we do know, and I have to say this is in part due to some of Avi's work. We do know that rocks from other solar systems do come whizzing through our solar system, and some of them do crash into planets here, including us. So, but one thing that we don't know is how common is life in the
Right.
universe, right? If life itself is something that's really hard to get going, it could be that even though there are millions of habitable planets in our galaxy, they might all be barren. We don't know. Half of the field of astronomy is dedicated to answering this question, right? By looking for signs, biosignatures, signs of life around on planets orbiting other stars, which is a wonderful and fascinating question.
Right. Well, I guess those drones weren't from other solar systems a month or two ago.
The what?
The drones, that appeared over
Oh,
in New Jersey and the rest of the country, people thought they
yeah,
were
right.
aliens.
Yeah.
Turned out not to. Maybe we'll find out now with new administration what those drones were all about. But anyway, I'd like to thank you very much for taking the time to explain mirror life to us and the potential significance and consequences of that if it's developed. It's important
Good.
that the public understand what
Yes.
it's about and could lead
Yeah,
to.
yeah, yeah. Thank you for helping us get the message out about this technology and yeah, what the potential problems are. Thank you.
And when you get that first microbe or bacteria that you develop in your lab, please contact me because I want to be the first one to interview you about it and your second Nobel Prize.
Okay, deal.
All right.
All
Take
right.
care.
Okay, you too. Thanks.